For over a millennium, medical science was stalled by inherited dogma. The Hippocratic humoral theory and Claudius Galen's anatomical deductions, based largely on dissecting macaques and pigs, ruled the academic world. Physicians attempted to treat imbalances of theoretical fluids rather than examining actual human physical structures. The Renaissance forced a break in this tradition. Scholars and artists began prioritizing direct physical observation over ancient texts to figure out how the body actually worked. In 1543, Andreas Vesalius published De Humani Corporis Fabrica. He abandoned animal proxies to perform first-hand human dissections, documenting the interior of the body with a level of detail that had never been committed to paper. By cutting into human corpses himself, Vesalius meticulously mapped the true material architecture of bones, muscles, and vessels, correcting centuries of Galenic errors. You cannot understand the mechanics of biological life without first defining its exact physical constraints. Careful, direct observation became the prerequisite for discovering physiological truth. Modern biology takes Vesalius's structural maps and sets them in motion. This integration relies on a central rule, the principle of complementarity. Form dictates function. The specific physical shape of a biological entity completely determines what it can achieve biochemically. Biologists adapted this framework from quantum physics. They realized that the spatial reality of anatomy and the dynamic biochemistry of physiology are simultaneous descriptions of the same organism. One provides the physical map, the other provides the live data. The human body builds itself across a strict structural hierarchy. Atoms bond into molecules. Molecules form cells. Cells weave into tissues, and tissues construct massive organ systems. Every level of this physical hierarchy is engineered to execute a specific physiological job. Anatomy is the physical geography. Physiology is the live traffic running through it. Neither can exist without the other. The cardiovascular system operates as a massive, integrated fluid transport network, tasked with continuously pumping oxygen and nutrients to trillions of cells. If you look at the gross anatomical structure of the human heart, you will find a glaring physical asymmetry between its two lower pumping chambers. This diagram shows a cross-section of the beating heart. The left ventricle possesses a remarkably thick, dense muscular wall, while the right ventricle is encased in a much thinner layer of muscle. The left ventricle must generate enough hydrostatic pressure to force oxygenated blood through the entire body's systemic circulation. The right ventricle only requires enough force to push blood a few inches to the adjacent lungs. When that left wall stretches and thins, a condition known as dilated cardiomyopathy, the physical engine fails. Without the structural density to generate high pressure, blood flow stalls, fluid backs up into the lungs, and systemic oxygen delivery drops. At the macroscopic level, the functional output of a biological engine is strictly limited by the physical integrity of its structural components. The same engineering laws that govern a beating heart dictate the behavior of individual microscopic cells. The digestive system faces a logistical problem. It needs to extract the maximum amount of nutrients from food and absorb them into the bloodstream, but it must do so within the confined space of the small intestine. The body solves this by folding the interior lining of the intestine over and over. These folds are covered in microscopic finger-like projections called villi, and the surfaces of those cells are modified into thousands of even smaller microvilli. By structuring the tissue with millions of tiny bristles, the intestine multiplies its surface area. This creates vast amounts of contact space for nutrient absorption without increasing the physical footprint of the organ itself. Whether you are examining the density of a cardiac chamber or the microscopic folds of an intestinal cell, structural complexity exists to stretch physiological limits. In the 19th century, French physiologist Claude Bernard observed that complex cells survive only when the internal fluid bathing them remains chemically and thermally stable. He called this the milieu intérieur. This stability is maintained through homeostasis, a state of dynamic, relative constancy of the internal environment. It is not a frozen state. Variables like temperature and blood pressure oscillate around a specific baseline, or set point. Homeostasis is governed by a three-part feedback loop. A specialized receptor detects a change, a central control center evaluates that data against the set point, and an effector executes a physical response to fix the imbalance. Most of the body relies on negative feedback loops. If your core temperature spikes, the brain activates sweat glands and dilates blood vessels, effectors that oppose the heat, driving the temperature curve back toward the baseline. A positive feedback loop works in the opposite way. Instead of opposing the change, the body amplifies the stimulus, pushing the variable further away from the baseline until a rapid, definitive endpoint is reached. In childbirth, physical pressure on the cervix triggers the release of the hormone oxytocin. Oxytocin causes uterine contractions, which pushes the infant harder against the cervix, releasing even more oxytocin. This cycle escalates until the infant is delivered. Biological survival is a continuous, active computation. The nervous and endocrine systems constantly adjust physical levers to counteract external environmental chaos. When these control mechanisms break down, the result is homeostatic imbalance. This is the physiological definition of clinical disease. Consider a patient experiencing a panic attack triggered by extreme anxiety and stimulants. The neurological stress triggers an immediate mechanical anomaly. The patient begins to hyperventilate, taking very rapid, shallow breaths. This overbreathing disrupts the gas exchange in the lungs. The rapid exhalations purge carbon dioxide from the bloodstream far faster than the body's metabolism can replace it. Carbon dioxide acts as an acid in the blood. As it vanishes, the blood's homeostatic buffer shatters, and the pH spikes into the alkaline zone. This state is known as respiratory alkalosis. That chemical shift causes a microstructural failure. The alkaline environment physically warps the shape of hemoglobin proteins. Instead of releasing oxygen to the tissues, the warped proteins tightly lock on to their oxygen molecules. This disrupts the Bohr and Haldane effects, the chemical mechanisms that normally allow hemoglobin to exchange gases based on local acidity and gas concentrations. The patient's lungs are pulling in oxygen, but their tissues are suffocating because the structural delivery mechanism has been jammed by the pH shift. The goal of medical intervention is to repair that broken feedback loop and restabilize the internal chemistry before the starving cells die. The biological rules of complementarity and homeostasis are permanent, but the tools we use to map them are expanding. For 500 years, medical students relied on physical cadavers. While dissection offers structural insight, a preserved corpse lacks the tactile reality and active feedback of a living, failing organ system. Today, medical education integrates spatial three-dimensional mapping and virtual reality. These digital ecosystems allow students to manipulate spatial relationships in real time, letting them dissect and rebuild complex neurological networks without any risk to a live patient. To master the human machine, modern medicine is leaving static textbooks behind in favor of interactive clinical environments. Diagnostic technologies like CT and MRI scans use geometric anatomical planes, transverse, sagittal, and coronal, to non-invasively map the interior of the living body. Algorithms now extract that raw radiological data to generate a digital twin. This is a highly accurate, dynamic 3D replica of a specific patient's unique anatomy, rendered from their own scan data. This data moves from the scanner to the operating room. Surgeons use the digital twin to carefully rehearse complex reconstructive procedures on a screen before making an incision. Clinicians can see exactly how a patient's individual structural geometry will react to a surgical intervention, reducing the risk of intraoperative complications. From Renaissance woodcuts to surgical simulations, the goal of medicine remains the same: mastering the link between human form and physiological function.